back to lecture number 2, cell the fundamental unit of life। तो आज हम क्लास 9th एनसीईआरटी का लेक्चर नंबर 2 देखने वाले हैं, जिसमें हम सबसे पहले बात करेंगे एक ओवरव्यू ऑफ सेल की, प्लाज्मा मेम्ब्रेन की और भी चीजों की। तो चलिए शुरू करते हैं। तो सबसे पहले ओवरव्यू ऑफ अ सेल कि भाई एक सेल में मोटा मोटा होता क्या है। वैसे तो सेल में बहुत सारी चीजें होती है, पर मोटा मोटी हर सेल में ये 3 चीजें होती हैं। न्यूक्लियस जो होता है वो किसी सेल्स में न्यूक्लियस नाम से होता है, तो किसी चीज में वो ना होके न्यूक्लियोइड होता है, न्यूक्लियोइड होता है। तो चलिए आगे बढ़ते हैं। तो ये जो आपको सबसे बाहर की कवरिंग दिख रही है, ये होती है सेल मेम्ब्रेन या प्लाज्मा मेम्ब्रेन। कुछ सेल्स होते हैं, जैसे एक प्लांट का सेल हो गया या फंजाई का सेल हो गया या फिर बैक्टीरियल सेल हो गया। इन सब में सेल मेम्ब्रेन के बाहर भी एक कवरिंग होती है, जिसे हम कहते हैं सेल वॉल। पर वो हर सेल में नहीं होती, वो बहुत ही सिलेक्टिव सेल्स में होती है। फिर आता है साइटोप्लाज्म। ये जो आपको पर्पल कलर से इसके अंदर दिखाई दे रहा है, ये वाला पूरा, ये साइटोप्लाज्म है। और ये डार्क पर्पल, ये जो बीच में गोला है, दिस इज नथिंग एल्स बट न्यूक्लियस। सो दिस वाज द ओवरव्यू ऑफ सेल। अब हम आगे बढ़ते हैं कि भाई प्लाज्मा मेम्ब्रेन होती क्या है। So, plasma membrane or cell membrane is a universal feature of a cell. It is present in each and every cell, whether it's of a animal, of a plant, of a fungi, of a algae, or any organism. It's a thin boundary. Plasma membrane is a thin boundary, and it's like surrounds and protects the cell content. It separates the cell content from the external things outside the cell and things inside the cell. Cell wall is basically selectively permeable, which means it allows some substances to move through it. Like it allows only very few substances to move in or out through it. So this was all about cell membrane. Now, and this everything I am discussing is from NCRT. It's like not out of it. Some things can be out of it, but mostly is from NCRT. NCRT का कुछ भी नहीं छूटेगा। हाँ, पर मैं थोड़ी बहुत चीजें बाहर की भी कभी कभी डिस्कस करती। सो आगे बढ़ते हैं। Now, structure of a cell membrane. As I told you, it's very thin. It's like 7 to 10 nanometers. 7 to 10 nanometers. It's made up of lipids and proteins. And if I'll tell you, so like very specific, if I'll go, it's not given in NCRT, but it's made up of phospholipids. It is made up of phospholipids. So it's not in NCRT, but I'm just telling you guys. So it's made up of phospholipids and some proteins. So phospholipid is nothing but is a kind of fat. Phospholipid. So protein acts as a gatekeeper, like with, as I told you, it's selectively permeable. So the substance which help in the transport, it's some proteins. Then fluid mosaic model of cell membrane. Nicholas and Singer were two scientists who gave this model. In this model, they told that plasma membrane or our cell membrane is made up of two layers. It is of two layers, right? Plasma membrane or cell membrane is made up of two layers. So plasma membrane is made up of two layers. So if you will see, so this is the first layer, this comes the second layer. So plasma membrane is like this. And if I'll tell you, so these are the tails of plasma membrane, and these are the heads. So tails are hydrophobic, they don't like water, they hate water. So that's why they're inside and they're safe from water. But the heads are water loving, water attracting, they're hydrophilics. That's why they're outside. So these are exposed to water, but these are not exposed to water as they're hydrophilic. I hope fluid mosaic model is now clear to you, and there's nothing in fluid mosaic model except this. So let's begin. Now if you will see, so fluid mosaic model, what is a fluid? What is a fluid? So fluid, the molecules move sideways. If these are the molecules, they'll move sideways, right? They'll move sideways and they'll make that thing flow. So whichever thing can flow is a fluid. And mosaic are like tiles arranged in a mosaic. Like tiles arranged to make something is known as mosaic. Right. For example, jigsaw puzzle. Like we arrange jigsaw puzzle. So it's a mosaic. It's an example of mosaic. So this is the basic idea of fluid mosaic model. Now let's continue with active and passive transport. So passive transport is basically when substance move to a higher to a lower concentration. Substance move from higher to lower concentration. For example, suppose this is a cell, right? This is cell membrane and this is uska pura concentration. Then this is a cell, right? So if you will see, so this is a cell, this is another cell. Like this is something which is concentrated, this is something which is concentrated. So the substance will move from higher to lower concentration to make things equal. To make things equal, right? To make things equal. Now they are equal. So this is what active transport is. Passive transport. What is active transport? Things move from higher to lower, from lower to higher concentration. Please correct this. So basically substance move from higher to lower concentration in active transport and from lower to higher concentration in active transport. In passive transport, there is no energy required, but in active transport, an energy is required which is known as ATP, adenosine triphosphate. The full form of ATP is adenosine triphosphate. This is a very slow movement, but this is a rapid movement. Only small molecules of water can be transported passively, but large molecules can be transported actively. I hope active and passive transport is clear to you. Now comes types of solution. So there are three types of solution. The very first one is hypertonic solution. In hypertonic solution, if you will see, so this is a beaker filled with hypertonic solution and this is a cell. What happens to cell? That cell shrinks. So in hypertonic solution, cell shrinks. if you will see, so this is a cell. This is a cell. I put it inside a hypertonic solution. Okay. I have put it inside. I have put it inside an hypertonic solution. Then what will happen? Cell will shrink. Cell will reduce its size. So, firstly, the cell was of 5 pens or 5 markers. Then it became of 2. So this is what hypertonic solution is. In hypertonic solutions, the content moves from higher to lower concentration. The concentration inside the cell is higher than the concentration outside the cell. That's why the water content or the materials inside the cell move outside and cell shrinks. So this is what when we put cell in hypertonic solution happen. So it will just shrink. Then in hypotonic solution, when we keep the cell in hypotonic solution, it swells. It don't shrink, it swells, because hypertonic solution is less concentrated than the cell. The If you will see, this is a cell. I put it inside a hypertonic solution. What will happen? It will shrink. But if I put the same cell in a hypotonic solution, what will happen? It will swell. Firstly, my cell was of 4 markers, then it became of 8 markers. So it swelled, it increased the size. This is what hypotonic solution is. Then in isotonic solution, cell remains same as the concentration inside the cell and outside the cell is the same. So both are same. That's why we don't we don't want any, we don't see any change in the shape or size of cell in isotonic solution. Now, what is diffusion? Diffusion is basically the movement of anything, like the movement of gases or liquids from higher to lower concentration. So this it is a spontaneous movement of substances from region to higher concentration to the lower concentration. Okay, if you will see here, so this is a high concentrated area. It is a high concentrated area. This is a low concentrated areas. So what will happen? These substances will move inside, and when it will be like same, right? Suppose this is a cell in hypertonic solution, right? And here diffusion takes place. So if you will see, this is the concentration inside the cell, this is the concentration outside the cell. So what will happen? So what will happen? Any guesses? No, I'll tell you. Not, if you will see, so firstly, if you will see, abhi bhi concentration outside the cell is more. So now it's same, this cell, and inside the cell, outside the cell, these both concentrations are same. This is known as diffusion. So, and now comes osmosis. Osmosis is nothing but the transfer of water molecules from higher to lower concentration through selectively permeable membrane. Now, osmosis can be of two types. It can be endoosmosis, it can be exoosmosis. Okay. Now, if you will see, so what is endoosmosis, what is exoosmosis? So endoosmosis is basically the movement of water molecules inside the cell. The word endo means inside. The word endo means inside, and osmosis means the movement of water molecules. So that is why endoosmosis is movement of water molecules inside the cell, and exoosmosis is the movement of water outside the cell. Exo means outside. Exo means outside. I hope this much is clear, that what is endoosmosis, what is exoosmosis. So now let's go ahead. So now what is the difference in diffusion osmosis? What is the similarity? So if you will see, so equalizes the concentration of two solutions. These both equalizes the concentration of two solutions. If there are two things, they'll equalize the concentration. This is what diffusion and osmosis both do. Now, diffusion is basically when solute or solvent move outside or inside the cell. There is no semi-permeable or selectively permeable membrane involved. But in osmosis, only solvent particles can move, only water particles can move, and a semi-permeable membrane is included in this, is involved in this. So this is what diffusion and osmosis difference and similarity is. Now this is for today, and then we'll meet in lecture number 3, in which we are gonna study cell wall, DNA, nucleus, and many more things. I'm very excited for that lecture. Tab tak ke liye bye bye, take care, and I'll meet you in lecture number 3. Bye 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 bye.